Good afternoon, everyone. My name is Heriberto Moran, and I'm going to talk about my project, which has to do with the regulation of hdm2 gene expression by hnrnpA1. And hnrnpA1 it's an RNA binding protein that plays a big role in gene expression. So the central question for my project is whether hnrnpA1 plays a role in the regulation of hdm2 gene expression. And as you know, gene expression can be controlled at multiple levels, at the level of transcription, at the level of post-transcriptional regulation, and also at the translational level. And it has previously shown that hnrnpA1 plays a role in at the level of post-transcriptional regulation. And hnrnpA1 consists, it's an RNA binding protein that plays major steps in the regulation of gene expression. For instance, it has been shown that it regulates the mRNA export and also at the mRNA stability. hnrnpA1 consists of an RNA binding protein motifs and also the G domain. And at the C-terminus, that's where the M9 region is located. In this region, it's important for the nucleocytoplasmic shuttling. And also, some of the structural features of hnrnpA1, it consists of 34 kilodaltons protein, and it can be modified after it's synthesized. And it is a member of the hnrnpA and B family of proteins, and it's involved, as I mentioned previously, in many aspects of pre-mRNA and mRNA metabolism. And it's also involved in the telomere biogenesis, and it's one of its main functions. It's in the regulation of alternative splicing, and also because it binds to the 3' UTR of mRNA, it's also involved in the mRNA turnover and translation. So what is the connection between hnrnpA1 and hdm2? Because we were interested in finding new targets of hnrnpA1, we found that it binds to hdm2 mRNA, specifically at this region called the intra-9 and exon 10 junction. And this was shown by a previous graduate student where she found that it was where she performed hnrnp co-IP immunoprecipitation with RNA extraction. And she found that it binds to this region. So what is hdm2? It is a gene. One of its main functions is that it regulates the p53, which is a tumor suppressor. And hdm2 consists of 11 exons and 11 introns. And its transcription it's initiated at two promoters, at the P1, which is p53 independent, and the P2, which is p53 dependent. It is a protein that consists of 491 amino acids, and it has a molecular weight of 56 kilodaltons. And also, hdm2 can be regulated at different levels, such as at the level of transcription, at the level of the post-transcriptional level. So again, one of its main functions of hdm2 is to regulate p53, and it does so by binding to its N-terminus. And this inactivates its transcriptional function of 53, and also it regulates p53. It degrades p53 by modifying by ubiquitinating p53, and then this induces the perosomal degradation, the degradation of p3 by the 26S proteasome. And in addition to p53, it binds to p14R. And this induces the sequestration of hdm2 away from p53, and this activates p53. So what do we know about A1? And from previous studies, we have found that it plays a key role in the regulation of gene expression. For instance, we have found that there are lower levels of hdm2A1, hnrnpA1 in senescent cells. And also when we overexpress A1, we found that there is a increase in the steady state mRNA levels of p14R and p16, which are tumor suppressors. And so this finding suggests that hnrnpA1 plays a key role in the cell proliferation and also in senescence. So my hypothesis is that hnrnpA1 regulates hdm2 gene expression by directly binding to hdm2 mRNA, and as a consequence of this, it affects its pre-mRNA, mRNA export, and stability. And it also involved that hnrnp is involved in the P14R53 hdm2 pathway. So my aim was to examine whether hnrnpA1 regulates hdm2 gene expression at the transcriptional level, subcellular localization, and mRNA stability. And what our previous preliminary data suggested was that when we overexpress A1, we found in using IMR90 cells, we found a downregulation of HDM2 mRNA levels. And when we silence hnrnpA1 with siRNA, we saw the opposite, that increased the levels of HDM2 mRNA. So in the next time, I'm going to go over more about my project, and I'm really thank you for taking your time to listen to my just a brief description of my research project. Thank you.